Hello guys, welcome back to my channel again. Today in this lecture we are going to talk about DNA replication. So in this complete lecture we are going to cover DNA replication, the process of DNA replication step by step in both prokaryotes as well as eukaryotes. So what is DNA replication? DNA replication is a process through which a single DNA helix can make an exact or identical copies of itself. So before going further into the process of DNA replication, let me tell you about the DNA. The DNA is a long polymer of deoxyribose nucleotide. See, these are the deoxyribose nucleotide, which is bond between two ribonucleotide. That's a phosphodiester bond between the 5' phosphide and 3' OH here, 3' and 5'. So the nucleotide in DNA are bonded together by actually phosphodiester bond at 3' and 5' carbon, not OH. So the DNA molecule has a direction. It has 5' to 3' direction. One end is called 3', the another end is called 5'. So the double strand DNA has a complementary strand run in opposite direction. If the one strand is 5' here and another strand would have 3' here in the exact opposite direction in double strand DNA. तो डीएनए रिप्लिकेशन किस में होता है? प्रोकैरियोट्स में भी होते हैं एज वेल एज यूकैरियोट्स में भी होते हैं. इन प्रोकैरियोट्स एंड यूकैरियोट्स. सो इन प्रोकैरियोट्स एक तो क्रोमोसोम्स में भी होते हैं क्रोमोसोमल रेप्लिकेशन एंड अनदर इज प्लास्मिड रेप्लिकेशन इन बोथ प्रोकैरियोट्स एंड यूकैरियोट्स. सो क्रोमोसोमल एंड प्लास्मिड रेप्लिकेशन. प्रोकैरियोट्स में भी यूकैरियोट्स में भी एक तो क्रोमोसोम्स में होते हैं मेन क्रोमोसोम्स एंड अनदर वन इज प्लास्मिड में होता है. अगर हम बात करते हैं क्रोमोसोम्स की इन यूकैरियोट्स, तो क्रोमोसोम्स एक तो न्यूक्लियर होते हैं, द अनदर इज एक्स्ट्रा न्यूक्लियर. एक्स्ट्रा न्यूक्लियर मींस जो न्यूक्लियस से बाहर हो. मेन न्यूक्लियस में जो जेनेटिक मटेरियल होते हैं उसका भी रेप्लिकेशन होता है. जो बाहर होते हैं ऑर्गेनेलीज में, फॉर एग्जांपल माइटोकॉन्ड्रिया में भी होते हैं और क्लोरोप्लास्ट में भी होते हैं. तो प्रोकैरियोट्स के जो क्रोमोसोम्स होते हैं वो जनरली क्या होते हैं? सर्कुलर होते हैं. और इनका जो सर्कुलर डीएनए का रेप्लिकेशन होता है उनको हम क्या कहते हैं? इट्स कॉल्ड थीटा रेप्लिकेशन. द प्रोसेस इज कॉल्ड थीटा रेप्लिकेशन. और जो प्लास्मिड में जो डीएनए होते हैं वो भी सर्कुलर होते हैं, पर प्लास्मिड डीएनए की जो प्रोसेस है कभी कभी उसको थीटा रेप्लिकेशन भी कहते हैं और कभी सिग्मा रेप्लिकेशन भी कहते हैं. एंड इन यूकैरियोट्स इज एग्जैक्टली सेम. बट आप लोग यहाँ पूछेंगे कि यूकैरियोट्स में किस में प्लास्मिड होता है? इट इज रेयर इन सम बडिंग यीस्ट सच एज सैक्रोमेसिया सर्विसिया में होते हैं. और यूकैरियोट्स के जो न्यूक्लियर क्रोमोसोम्स होते हैं इट इज एक्चुअली लीनर होता है और इसका जो रेप्लिकेशन होता है वो सेमी कंजर्वेटिव इन नेचर होते हैं. तो लीनर फॉर्म ऑफ डीएनए होते हैं. और जो प्लास्मिड होते हैं वो भी सर्कुलर ही होते हैं और इट टेक्स प्लेस थ्रू द प्रोसेस ऑफ थीटा रेप्लिकेशन. एंड इन एक्स्ट्रा न्यूक्लियर सच एज माइटोकॉन्ड्रिया एंड क्लोरोप्लास्ट इट हैपेंस एज ए डी रेप्लिकेशन, या इसको हम क्या कहते हैं? डी रेप्लिकेशन कहते हैं और डिस्प्लेसमेंट रेप्लिकेशन कहते हैं. So the introduction of DNA replication. DNA replication is actually the basics of inheritance. Inheritance, which means the transferring of genetic material from parents to offspring, or any character or any gene from parents to transferring of genes from parents to offspring is called inheritance, or any characters. So DNA replication is a fundamental process which occurs in all cells for copying DNA to transfer the genetic information to the offsprings or the daughter cell. So DNA replication happens when the cells are divided. Each cell must replicate before division. So the number of chromosomes, so the number of amount of DNA remain same in our cell. DNA replication is actually the semi-conservative in nature. Semi-conservative means the strand. There are two strands of DNA. One strand remains same in all one strand remains same in all the step of in all the process in all the process of DNA replication. One is template strand, another strand is so the parental strands are not degraded, it remains till the end. So the base pairing allow each strand to serve as a template for new strand. So this one strand becomes a template to formation to synthesis a new strand of DNA. So the new duplex is half is the original, half is the new one. For example, this one is new and this one is the original strand. See in this process, in this picture, you can see this is the original DNA, and when there is a replication, one normal copy becomes another copy is here. So the DNA replication is actually called semi-discontinuous process. Why it's called semi-discontinuous process? Because the leading strand and lagging strand. The leading strand is continuously synthesis from 5' to 3' direction, and the lagging strand is synthesis in discontinuous process in short fragments. So the leading strand is continuously synthesis or continuous strand, or the lagging strand is also due through the synthesis through the Okazaki fragments, which are joined by ligase enzyme. See in this picture, you can see this is the template DNA, original DNA. This is the replication fork, and this is DNA polymerase, which is the main enzyme responsible for DNA replication, and this is the new strand, leading strand, which is continuously synthesis from this 5' towards the 3', and this is another strand, this is the replication fork, and this is another strand of DNA which is getting synthesis in short fragment. This is DNA polymerase, and this DNA ligase will ligate both the strands and make a continuous at the end. These are Okazaki fragments. So sometimes Okazaki fragments are also called as the hybrid of RNA and DNA, because there is RNA primer at the each and every between this, and these primers are being removed at the end, and the ligase can bind these small fragments of DNA into long continuous strand. So DNA replication needs energy. So the energy for DNA replication arrived from nucleoside triphosphate. So the sugar with triple phosphate triphosphate. So between this phosphate there is energy. When this bond breaks, it releases energy. So between two phosphate groups, the phospho in a hydride bonds are there, and they are highly energetic bond, and when these bonds break, it release energy. So the DNA bases arrive with their own energy source for bonding. These are bonded by enzyme DNA polymerase. Third, DNA replication needs primer. Primer is what is primer? Primer is synthesized by primerase, the RNA primer mainly. Primer is a small fragment. Primer is a small fragment which starts the DNA replication with a small fragment, because the DNA polymerase only recognize 3' only need 3' to grow. The limitation is 3' because and the DNA primerase synthesis this short fragment primer of 5 to 3 nucleotide or 5 to 6 nucleotide with 5' to 3' and without any template. So this is primer, and the DNA replication or DNA polymerase starts the whole process of replication or synthesis the long string from this 3'. So the template is read in the 3' 5' direction, while polymerization takes place in 5' to 3' direction. So that's the need of primer. So primer synthesis by primerase. Primerase is the enzyme which synthesis actually RNA primer, and it serves as a starting or sequence for DNA polymerase third. So because DNA polymerase third do not synthesis or do not add nucleotides without the 3' of the DNA, so only one RNA primer required for the leading strand, but the lagging strand Okazaki fragment, it needs the number of fragments or depends on the Okazaki fragment are number of primers. So RNA primer has a free 3' OH. From these 3' OH, the replication process is going to continue, or synthesis or edit of nucleotide happens. So these 3' OH are the primer is the first nucleotide which is born. So DNA replication steps. The steps of DNA replication is, the first is identification of the origin of replication. So origin of replication in prokaryote is ORIC, and eukaryote is ORA, something like that. So the second step is unwinding of the DNA. Unwinding means denaturation. See, the DNA look like this, and before replication, it's need to be unwind like this. So for example, when these bonds, there are bonds between two nucleotides, there is hydrogen bond between adenine and thymine, guanine and cytosine. So the bonds are break and the DNA are actually denatured and it unwind and it provide a single strand template. It is double stranded DNA. So after unwinding it, it makes a replication fork look like this. The replication fork look like this. For example, see, this is the replication fork. For example, this, it becomes single strand in here. So the initiation of DNA synthesis and elongation start from this origin of replication. So the primer removal and ligation of the newly synthesized DNA fragment at the end after elongation. So the reconstitution of chromatin structure takes place. Component of DNA replication. DNA polymerase is the main enzyme which is needed for DNA replication. Deoxynucleotide polymerization is also called as DNA polymerase, which is the main enzyme. It has number of types like DNA polymerase first, second and third. Fourth, and helicase, which is processive unwinding of DNA. Unwinding of DNA need an enzyme which is called helicase. And topoisomerase is another enzyme which relieves the tension or transitional strands that results from helicase induced unwinding. And RNA primase is the enzyme which makes the RNA primer during the initiation of this replication, and single standard binding protein, some protein which prevent premature re-annealing of DNA synthesis. SSB protein is also called SSB protein. So after that, the nick formation which happens due to the removal of primer like this. For example, this is the primer and DNA replication has been started, and another primer is here and another Okazaki fragment comes here, and after removing these primers it look like this, and these nicks are connected or ligate by an enzyme called DNA ligase, which which seal the nicks between the nascent chain and Okazaki fragment on lagging string. The origin of replication in prokaryote, as I wanted to say in the first, the first slide, and the origin of replication in prokaryotes are E. coli, AIDS, or I. See, there is an association of sequence-specific double-stranded DNA binding protein, which is a series of direct repeats DNA sequences. So, in E. coli, the protein of DNA protein, which is used for a complex which is formed. Complex which is formed is a monorepliconic, which is single origin of replication in prokaryotes, and these are cis-acting elements, and it is approximately 245 base pairs, and they are AT rich, adenine thymine rich. This region are because the denature and unwinding of an addition AT is very easy, because adenine and thymine is only double bond as compared to the guanine and cytosine, which have triple bond. And there is another one repetitive sequence which is called 13-mer in prokaryotes, or another one is GATA sequence, GATC. You can say got a sequence and this 13-mer, or another one sequence is 9-mer. So the replication or mesheting of the DNA starts from these sites. Origin of replication in eukaryotes is functionally similar or autonomously replicating sequences. It is also called as replicators. It has been, identified in budding yeast. The origin of replication in eukaryotes contains somewhat degenerated almost 11 base pair sequence called it origin replication element, which is called it ORE. The ORE binds to a set of protein which is analogous to DNA in prokaryotes, which is collectively called as origin recognition complex ORC. The ORE is located in the adjacent to an approximately 80 base pair 80 rich sequence that is easy to unwind. This is called the DNA unwinding element. This 80 base pair sequence is called DNA unwinding element, which can help in unwind the DNA on the 80 rich sequences. So unwinding of DNA is the interaction of protein with ORI defines the start site of replication and provides a short region of single stranded DNA essential for insertion of synthesis of the nascent DNA strand. See, this is the DNA. It's going to unwind. See, for example, it unwinds here by helicase enzyme. See, this is helicase enzyme. It start unwinding the DNA, and after that the SSB proteins, which is in tritrameric forms, it binds here, so the unwinded DNA do not get wind again due to the strain which is caused here. See, if we unwind here, अगर हम यहाँ पे unwind करते जाएगा, यहाँ पे सीक्वेंस में क्या आ जाएगा, स्ट्रैंड आते जाएगा और इसको टोपोआइसोमेरेज रिलीज करते हैं स्ट्रैंड. In cooperation with SSB, this leads to DNA unwinding and active replication. This is the process of unwinding. See, helicase enzyme unwind this DNA, and after that the SSB protein will attach here like this, and this is called the replication from where the after that the DNA polymerase enzyme will bind here and start the replication process. So formation of replication from the polymerase third hollow enzyme which bends the complex DNA as a part of multi-protein complex. So जो DNA polymerase third होते हैं वो उसके बहुत सारे क्या होते हैं parts होते हैं, इसलिए इसको हम क्या कहते हैं multimeric protein. So DNA polymerase only synthesize DNA in 5 days to 3 days direction, as I already said. So the DNA strands क्या होते हैं? Actually they are antiparallel. The polymerase function asymmetrically. On the leading strand or forward strand, we can say the DNA synthesize it continuously, which is continuous strand, leading strand, and on the lagging strand, which is opposite in this direction, the DNA synthesized in short 1 to 5 kb fragments, the so-called Okazaki fragments. See, this is the structure where the DNA replication takes place. This is replication form ultimately. So this is the parental DNA. Helicase will unwind from here, and these are SSB proteins which helps in keeping this DNA unwind, and this is the DNA polymerase which synthesis. See, this is the leading strand which synthesis from. This is RNA primer here which synthesis the DNA in the 5 days to 3 days direction. Same here in Okazaki segment is exactly opposite. This is primerase which is primer and start the replication here. These are Okazaki fragments. These are again primer, and this is in opposite direction because DNA has bidirectional replication. Overall direction of replication is from 5 days to 3 days direction, we can say. Formation of replication bubbles in eukaryotes, because prokaryotes has only one origin of replication, but in eukaryotes they have the number of replication fork. It occurs number of replication fork. Replication occur in both directions along the length of DNA in both strands are replicated simultaneously. So I said in the previous slide also, this replication process generates replication bubbles. See, look like this. These are replication bubbles. There are number of replication forks takes place in the same DNA, and this is the daughter DNA or the synthesis of new strands takes place. The DNA polymerase complex is the number of DNA DNA polymerase molecule engage in DNA replication. They share the important properties, which is chain elongation, porosity, and proofreading. What is chain elongation? It is the account for the rate in nucleotide per second at which polymerization occurs. And porosity is an expression of the number of nucleotides added in the messenger chain. कितना न्यूक्लियोटाइड वो ऐड करके उस चेन को लंबा बना देते हैं. And proofreading is the function identifies copying error and correct them कि कहीं गलती से A के जगह पे T तो नहीं आएगा, T के जगह पे G तो नहीं आएगा, उसको देखने के लिए proofreading. So DNA polymerase complex in E. coli DNA polymerase third is called main replicase or main enzyme for responsible for DNA replication. Its function is to at the replication of all our polymerase. It has the highest rate of elongation as well as highest porosity. And DNA replication or DNA polymerase second is the most involved enzyme which helps in proofreading and DNA repair. And polymerase 1 is a complete chain synthesis between Okazaki fragments and the lagging strand. The difference between DNA polymerase first, second and third is the polymerase first have low polymerization rate and second is also low, and the high polymerization rate is DNA polymerase third. That's why all these activities, all these properties of DNA polymerase third which it have make it the main replicative enzyme or main replicase. And the porosity is also low in first and second DNA polymerase, and DNA polymerase third is high porosity, कितने number of nucleotide ये add करते हैं. And proofreading is 3' to 5' and 5' to 3' exonuclease as well as endonuclease. both होते हैं इसमें exonuclease proofreading के लिए. DNA polymerase 1 में DNA polymerase second have only 3' to 5' exonuclease activity, and DNA polymerase third have also 3' to 5' exonuclease activity. So primer removal, DNA polymerase helps, and primer removal is the best enzyme to remove primers, and the DNA polymerase second and third do not have these characters. And the strand synthesis lagging strand, it only helps in lagging strand, and the DNA polymerase this seal the nicks in the lagging strand, and DNA polymerase second have no role, and DNA polymerase third is the main replicase enzyme which synthesis the strand in both strands. And DNA repair does by both DNA polymerase first and second and third do not have any role. So in eukaryotic DNA polymerase, the eukaryotic cells have counterparts of each of these enzymes plus some additional ones. Comparisons which are shown here is like DNA polymerase 1 in prokaryotes. In eukaryote there is alpha polymerase which helps in gap filling and synthesis of lagging strand, and DNA polymerase second in prokaryotes which is exactly like the epsilon polymerase in eukaryotes, and it helps in proofreading and DNA repair. There is one another protein which is beta polymerase which help in DNA repair, and gamma mitochondrial DNA synthesis. These are the enzymes of DNA polymerase third like enzymes in eukaryotes. And DNA polymerase third like enzyme in the eukaryote is delta polymerase which has high porosity as well as the leading strand synthesis. Incision and elongation of DNA strand. So primer is the first step, the priming process, which involves the nucleophilic attack of 3' hydroxy group in the RNA primer on the phosphate of the first entering deoxynucleoside triphosphate with the splitting of pyrophosphate. ये वहां से एक pyrophosphate को रिमूव करके वहां पे हाईली क्या फॉर्मेशन करते हैं, न्यूक्लियोफिलिक अटैक फॉर्मेशन करते हैं, तो वहां पे न्यूक्लियोटाइड अटैक होकर वहां पे एट हो जाते हैं. सो इन मेमेलियन और यूकैरियोटिक डीएनए पॉलीमरेज अल्फा which is mainly responsible for the synthesis of primerase in eukaryote, but primers in eukaryotes, but in prokaryotes the primase enzyme is responsible for synthesis of RNA primer. So the selection of the proper deoxynucleotide whose terminal is 3' or 5', which just depend upon the proper base pairing with one strand to another strand of the DNA. So it goes according to the Watson and Crick, like A will always bind with double bond with T, and G is double bond with triple bond with C. It's like this. So this is the base pairing in RNA replication. See, this is the first original DNA strands. This is the unwinding steps, unwinding DNA replication. This is the new strands which is formed here, and it's base paired with A with T, G with C. The function of DNA topoisomerase enzyme, which is relief the supercoiling, is done by topoisomerase. It's of two types. One is topoisomerase 1 and topoisomerase 2. Topoisomerase second is also called gyrase. The topoisomerase 1 acts by making a transient single cut in the backbone of DNA, enabling the strand to swirl around each other to remove the build-up of activist C. अगर हम DNA को यहाँ से निकालते जाते तो यहाँ बहुत क्या हो जाता है, ट्विस्ट हो जाते हैं डीएनए यहाँ पे। तो इसको रिमूव करने के लिए, इसको खत्म करने के लिए डीएनए टोपोइसोमरेज मदद करते हैं। वन हमेशा सिर्फ एक साइड को ही कट करके इसका वो स्ट्रेन जो है रिलीज कर देते स्ट्रेस, लेकिन टोपोइसोमरेज सेकंड दोनों डीएनए पे, दोनों स्ट्रेन्स पे कट मारते हैं और ये अनवाइंड हेल्प करते हैं डीएनए टू पास थ्रू अनदर। So this is the formation of supercoiling. This is how here the supercoils happen when the DNA strand is separated one another by helicase, and this is called positive supercoiling. And that's how the topoisomerase 1 release this strand and seal the nick. And this is how the topoisomerase second works, which forms a nick in both the strands. Primer removal and nick sealing. So the primer was added during the process of the process of replication at the starting point. This primer is removed by DNA polymerase 1 by replacing ribonucleotide with deoxyribonucleides. So nicks are sealed by DNA ligase, and multiple primer can be ligate strand with single primer on the proofreading and editing is done by DNA polymerase first as well as DNA polymerase second, which can see the mismatch and abnormal bases are added and they are removed and repaired the system. So termination of replication is the process. Is completed in about 30 minutes, and replication rate is 3 into 10 to the power 5 base per 5 minute in prokaryotes. So the entire mammalian genome replicates in approximately 9 hours. The average period which is required for the formation of tetraploid genome from the diploid genome in a replicating cell. So the termination of replication is the process in 1 minute. Reconstitution of chromatin structure helps at the end by assembling the DNA synthesis with histone and the DNA synthesis and the cell cycle. So we all know that cell cycle G1S, G2MS is where the DNA replication takes place, but the starting of this replication is started in G1 phase of the mitosis and G2 phase. So telomerase in eukaryote, the DNA gets shorter and shorter at the end because of the lack of 3-OH after removing the primer at the end. This is called telomere, and one enzyme which is called telomerase, which helps in making your 3' this telomerase and making this short span along. This is repeats on the telomerase sequence using an internal RNA templates. This is the mechanism. These are some inhibitors of DNA replication, such as the gyrase topoisomerase second, inhibited by novobiosins and naladixic acid, ciprofloxacins, which is an inhibitor of DNA breakage and rejoining process, and many other drugs which helps in inhibiting DNA replication. So this is the entire or exact summary of the DNA replication. What is first is unwinding, second is primase, and third is continuous synthesis, and fourth is discontinuous synthesis, and at the end that is proofreading. So unwinding from the replication from primase synthesis the RNA primer, and the leading strand which is continuously synthesis the strand, and the discontinuous synthesis lagging strand which is occasional segment synthesis, and always in 5' to 3' direction, because DNA replication need 3' free 3' OH, and proofreading And after this, the primer is removed and nick is sealed by ligase and proofreading done by DNA polymerase second, and organization of the chromatin structure again as a chromosome. Thank you for watching till this, and if you like the lecture, please share it with your friends and subscribe my channel. Thank you so much.